Welcome to the Deep Dive. We take a stack of sources, research papers, articles, notes, and really unpack them to find the key insights for you. Today, we're diving into a specific scientific paper. It's from the journal Chemico-Biological Interactions. Our mission here is, well, to crack this thing open, understand what it found about two substances you likely encounter, maybe daily, and figure out, what it might mean.
Right. And the paper focuses on polystyrene nanoparticles, or PS-NPs, think really, really tiny plastic bits, and fluoride, both common, like you said. PS-NPs are, well, they're everywhere as pollutants, and fluoride is found naturally, plus it's often added to drinking water. So this study looks at their impact specifically on human colon cells, what happens when cells meet them alone, and crucially, what happens when they meet them together.
Yeah, and here's what grabbed me right away. The authors say basically that this is the first study they know of that specifically looks at the interaction between these plastic nanoparticles and fluoride. In terms of toxicity, okay, so let's unpack that. Let's start with the why. why these two? Polystyrene nanoparticles and fluoride. And why specifically colon cells? Well, polystyrene, it's ubiquitous, right?
Yeah.
Used in absolutely massive amounts globally.
Oh, yeah. Tens of millions of metric tons every year in North America, Europe, Asia. Huge numbers.
A lot of it's single-use stuff, too.
Exactly. So it becomes pollution incredibly easily, breaks down into smaller bits, microplastics, then these even smaller nanoparticles they studied, they get into water, soil, they can build up in the food chain.
So these PS-NPs are kind of a model for plastic pollution in lab studies because polystyrene is so common.
Precisely. And think about exposure through the environment, obviously. Contaminated water, food, even migration from plastic packaging. The paper mentions like 80,000 tons of polystyrene food packaging just in the U.S. each year.
Wow, okay. And the main way we encounter this stuff, and fluoride too, is through ingestion, right? Eating and drinking.
Yeah.
So the gut, the gastrointestinal tract, is a key entry point, which makes studying colon cells at the end of that tract seem pretty logical.
Makes perfect sense. Now add fluoride to that picture. It's naturally there in soil, water, some foods, tea, leafy greens, fish, and of course it's deliberately added to water supplies, toothpaste, for dental health.
Right, prevents cavities.
It does. Beneficial at low levels, for bones, teeth. But too much fluoride, that's known to be toxic. High levels are linked to things like DNA damage, oxidative stress, problems with the liver, kidney, brain.
The WHO has guidelines, doesn't it?
Yeah, they recommend around 0.7 milligrams per liter, not over 1.5. But the paper points out some places have way higher levels, like 22 milligel in one area in China mentioned. That leads to serious health problems, endemic fluorosis affecting bones, teeth.
Okay, so how does ingested fluoride get to the colon?
Well, about a quarter is absorbed in the stomach, but the rest moves into the intestines. So yeah, it's entirely plausible that your colon cells could encounter both these nanoparticles and fluoride at the same time as they pass through.
That potential for co-exposure is really the key here.
Exactly.
So the core question the researchers set out to answer was, just how toxic are these specific polystyrene nanoparticles to human colon cells in a lab dish, in vitro? And critically, does adding fluoride into the mix change that toxicity?
And to figure this out, they used a very specific type of PS-NP, amino-modified, about 100 nanometers. They carefully measured their physical traits, size, charge, all that. Standard stuff, but important.
To know exactly what they were working with.
Right. Then they took 3 different human colon cell lines. These are like standard models used in labs and exposed them. Different amounts of nanoparticles, sometimes with fluoride, sometimes without, for different lengths of time, up to 72 hours.
And they didn't just see if the cells died, right?
No, much more detailed. They ran a whole battery of tests, measuring cell health, stress markers like ROS, whether the cells could divide properly, if they were undergoing programmed cell death, apoptosis. They even used powerful electron microscopes to actually see if particles got inside and if they caused, visible damage.
Which raises a big question straight away, doesn't it? How do these lab conditions, these specific concentrations, relate to what humans might experience over long periods at lower levels?
That's always the question with in vitro studies. A crucial first step. But yeah, translation to the real world is complex.
Okay, so what do the particles look like physically?
The paper describes them as tiny spheres, about 96 nanometers when dry.
Tiny.
Very. When they put them in liquid, like the cell culture media, simulating being near cells, their effective size, the sort of hydrodynamic diameter, was a bit bigger, around 120 nanometers, includes the water layer around them.
Okay.
And get this, when they added fluoride to that liquid, the nanoparticles seemed to swell up a bit, got about 15–20% bigger, closer to 130 nanometers.
Huh. Interesting. Did the fluoride change their charge?
Apparently not much. Their surface charge stayed negative, around minus 30 millivolts, with or without the fluoride.
All right, so let's get to the biology. What happened to the cells? Starting with cytotoxicity, how toxic were they?
Okay. For the PS-NPs alone, looking at the main cell line they used, HT29, toxicity depended on dose and time. Didn't see much effect at 24 hours, but by 48 and especially 72 hours, the higher concentrations, 250 and 500 micrograms per milliliter, definitely reduced cell viability. They were cytotoxic.
And the fluoride by itself?
Interestingly, at the concentration they tested, up to 1 millimolar, the fluoride alone was not toxic to these HT29 cells in this particular study.
Okay, so PSMP is toxic at high doses long times, fluoride not toxic at this level. What about different cell types?
Yeah, they checked other colon cell lines too. Seemed like some were more sensitive to the PSNPs alone than others. CACO2 cells were most susceptible, CCD841-con most resistant, HT29 somewhere in the middle, standard variation between cell lines.
Right, now the big one, the combination, PSMPs plus fluoride.
So when they looked at co-exposure, say 500 micrograms per milliliter of PSNPs with 1 millimolar fluoride for 72 hours, they did see a significant cytotoxic effect. Cell viability dropped compared to untreated cells. But, and this is important, the paper notes that this drop in overall viability wasn't, quote, prominent if compared to the cytotoxicity of PSNPs alone across all the cell lines at that specific high dose and time point.
Wait, so the combination wasn't dramatically more deadly overall than the nanoparticles alone? That seems unexpected, given the whole point was interaction.
It does seem a bit counterintuitive at first glance, looking purely at that overall viability number at that one condition.
But they dug deeper, right? Looked
Yeah.
inside the cells. You mentioned ROS, reactive oxygen species, stress signals.
Exactly, and this is where it gets really interesting, because surprisingly, in this study, neither the PSMPs alone nor the combination with fluoride significantly boosted ROS production in those HT29 cells.
Huh. So oxidative stress, which is often a go-to explanation for particle toxicity, wasn't the main story here, or at least not strongly induced under these specific conditions.
It suggests that, yeah, for this specific setup, these particles, this concentration, this fluoride level, maybe oxidative stress isn't the primary driver of the damage they observed, which leads to, well, what is going on?
Okay, so if not ROS, what else did they look at? The cell cycle, how cells divide.
Yes, and here the combination effect was much clearer. The PSNPs alone, even at the high dose for 72 hours, didn't really mess up the cell cycle distribution significantly. Cells were kind of progressing through division phases normally.
But with fluoride added.
But with the fluoride present alongside the PSNPs, they saw a distinct shift. More cells were getting sort of stuck or paused in the early GOG1 growth phase, and fewer cells were making it to the G2M phase, which is where they prepare for and actually divide.
So the combination was stopping cells from dividing properly? That seems significant, like jamming the cell's reproductive machinery.
It definitely suggests an interference with a fundamental cell process. It's not just killing them outright, maybe, but stopping them from functioning and multiplying correctly.
Okay, halting division. What about triggering self-destruct? Apoptosis. Programmed cell death.
They looked at that too. PSNPs alone did push some cells towards apoptosis. Specifically, they saw an increase in early apoptotic cells.
In the combination.
The co-exposure, PSNPs plus fluoride, increased both early and late apoptotic cells. The paper actually uses the word enhanced compared to PSNPs alone.
Ah, okay. So even if the overall number of dead cells wasn't drastically higher in that one snapshot, the combination was more effective at pushing cells further down that specific pathway of programmed death.
That seems to be the implication, yes. A stronger push towards apoptosis.
Now, for any of this to happen, the particles presumably have to get inside the cells, right? Did they check that?
They did. They saw increased cell granularity, which is often an indicator that cells are taking stuff up, internalizing things. And then they used TEM transmission electron microscopy, super powerful microscope. And yes, they could actually see the nanoparticles inside the HT29 cells.
Wow. What did it look like in there?
At lower PSNP concentrations, the particles were inside, but the cell structures looked mostly okay. But at the higher concentrations, the ones causing the toxicity. More nanoparticles inside, definitely. And signs of stress within the cell, increased vacuolization, like little bubbles or sacs forming, and damage to the mitochondria.
The cell's power plants.
Exactly. And crucially, these signs of internal damage, these ultrastructure changes, were even more prominent when the cells were exposed to the PSNPs with the fluoride.
So the fluoride seemed to worsen the internal disruption caused by the particles getting inside.
That's what the visual evidence suggests. Yes, it supports the idea that internalization happens, it causes stress, and fluoride might exacerbate that internal impact.
Okay, let's try and pull these threads together from the results section.
Okay, so PS-NPs alone are toxic, concentration, and time dependent. Fluoride alone at this 1 millimole level wasn't. The combination, well, it significantly reduced viability compared to control, but maybe not drastically more than PS-NPs alone overall at that specific high dose time.
Oh, yeah.
But the combination had clearer, enhanced effects on specific processes. It significantly disrupted the cell cycle, stopping cells from dividing properly, and it significantly enhanced apoptosis, pushing more cells into programmed death. And the internal cell damage seen under the microscope was worse with the combo.
So it's less about just killing more cells outright, and maybe more about changing how the cells are being damaged or pushed towards death, interfering with specific pathways more strongly.
That seems to be a fair interpretation of their findings, yeah. The nature of the toxicity seems altered or certain mechanisms amplified by the fluoride's presence.
Right. So how did the paper's discussion section frame all this in the bigger picture?
Well, they reiterate their main finding. PS-NPs are cytotoxic to colon cells that get inside, and fluoride enhances certain effects, particularly disrupting the cell cycle and boosting apoptosis. They also, importantly, acknowledge that studies on PS-NP toxicity are kind of all over the place.
Why is that?
Just depends so much on the specifics. Particle size, surface charge, surface modifications like the amino modification these had the type of cell used, how long the exposure is. All these variables can lead to different results. You can't just say, polystyrene nanoparticles are X toxic. It's nuanced.
Did they mention anything specific about the amino-modified ones they used?
Yeah, they noted that other studies have found these amino-modified PS-NPs can be more cytotoxic than some other types and potentially interact with mucin, which is the main stuff in gut mucus.
Ah, interesting. Protective layer interaction.
Could be relevant.
Yeah.
They also touch on mechanisms other papers have found, like oxidative stress, mitochondrial damage, activating the inflammasome, even though the study didn't find a strong ROS signal under their conditions.
Right.
They mentioned something called the proton sponge hypothesis as a possible way. Charged nanoparticles like these could trigger apoptosis by messing with lysosomes inside the cell.
Okay. And what about the fluoride context again?
They reiterate that while their 1 millimolar concentration wasn't toxic here, other studies definitely show fluoride toxicity at higher levels, usually over 4 millimolar. And the known mechanisms for fluoride toxicity, oxidative stress, DNA damage, mitochondrial issues, there's some overlap with potential nanoparticle effects.
But the really novel part was the interaction, right?
Absolutely. They emphasize how rare it is for studies to look at interactions between xenobiotics, foreign substances. It's complex, harder to do, and they point out fluoride is already known to make other things worse, metals, pesticides, some drugs. And, very relevantly, fluoride has been shown to increase the toxicity of silver nanoparticles in other studies.
So there's a precedent for fluoride interacting with nanoparticles.
Exactly. And they also mention that for aquatic life, PS-NP toxicity can be worse when other pollutants like pesticides or metals are present. So this idea of combined effects isn't totally out of the blue.
Okay, so connecting the dots for us, the listener, what's the main takeaway here?
I think the core message is this. This study shows pretty clearly in this lab model that even if one substance, like 1 millilem fluoride, isn't causing obvious harm on its own, its presence can significantly change how another substance, like these PS-NPs, affects cells. Specifically, it amplified the disruption to the cell cycle and pushed more cells towards programmed death. It really underscores why looking at pollutants in isolation might not give us the full picture.
Yeah.
Combinations matter.
Right. It confirms that stuff found together, like potentially in our water or food, can have interactive toxic effects, at least in this kind of lab setting. So let's bring it back to you, listening. We've gone deep into this one study on PS-NPs, fluoride, and colon cells. The key takeaway seems to be nanoplastics alone can be bad for these cells. Adding fluoride, even at a level not toxic by itself here, didn't necessarily kill way more cells overall in their snapshot, but it did enhance specific negative effects, messing with cell division and pushing cells towards self-destruction.
Yeah, and look, this isn't about causing alarm. It's about understanding the complexity, the environment, our food, our water. It's all a mixture. This study is a piece of the puzzle showing how those mixtures might behave differently than you'd expect just by adding up individual effects.
Absolutely, and we have to keep repeating. This is in vitro. Lab dish. Cell lines, it's an essential step, gives us clues, but it's not the same as a whole complex human body.
Definitely not. Digestion, metabolism, the immune system, the gut microbiome, so many other factors come into play in a living person.
But it does raise some really fascinating questions about these combined exposures, doesn't it?
It really does. It makes you wonder. Given we're exposed to countless combinations of chemicals and particles every single day, how many other interactions might be happening that we just haven't even thought to look for yet?
That's a powerful thought. And maybe a final one to leave you with, drawing straight from the study's implications.
Yeah.
If common substances found together can enhance each other's negative cellular effects in the lab,
Yeah.
what might that mean for how we assess chemical safety in the real world, where exposure is always to a complex mix, not just one thing at a time?
Yeah. How do we account for those potential synergies or enhanced effects in our risk assessments?
Something to definitely mull over.